Hello everyone, my name is Garhima Arora and I am a PhD student at Translational Health Science and Technology Institute, India. The topic of my presentation is Emergent Metabolic Landscape in the Transitory Ovarian Cancer Cell Niche Revealed through Genome-scale Metabolic Modeling. Before going into the details, I would like to give a brief description about the background. Ovarian cancer is one of the most common gynecological malignancies worldwide, which is associated with poor prognosis and a high mortality rate. Global estimates have revealed that the incidence of ovarian cancer among other cancers in female is around 3.4% with less than 50% 5-year survival rate. Due to delayed onset of clinical symptoms in ovarian cancer results in more than 70% of the cases left undiagnosed until the cancer reaches its advanced stage. The ovarian cancer metastasis starts with the detachment of the transformed cells from its primary tumor and exfoliating into the peritoneal cavity where they form the spheroids and these spheroids float and attach themselves to the secondary sites where they form metastatic tumor. A recent study by Langthasa et al. Demonstrated the morphological transition during ovarian cancer. In this study, they have used OVCAR3 cell line and they cultured for 24 hours and they observed that the single cells aggregated into the dysmorphic solid cluster known as morelloid spheroids. And when these morelloid were further cultured, they combined to form bigger clusters known as blastoid with a lumen-like structure. Now, the formation of spheroids is highly associated with drug resistance, disease recurrence, poor overall survival and metastasis. And many previous studies have represented significant findings demonstrating the metabolic alteration during ovarian cancer progression, including perturbed pyruvate metabolism and increased dependency of ovarian cancer cells on fatty acid for their survival. To best of our knowledge, there are no studies examining stage or morphology specific changes in metabolic dynamics of ovarian cancer. So therefore, this current study aims to capture the changes in metabolic landscape during phenotypic transitions and simultaneously targeting them to halt the disease progression. The current work is a combination of in silico as well as in vitro framework. Therefore, to accomplish our aim, we have used human genome scale metabolic model Recon 3D. Now, these models are computational framework that describes the complete generalized metabolic network. They mathematically represent the metabolic reactions which are carried out in an organism. In our study, we have used OVCAR3 cell line and have cultured 2D and 3D. Then high throughput proteomics was performed in the study to acquire the protein expression data. The protein expression data thus acquired contains the protein expression of around 3100 proteins across 9 samples, which are 3 biological replicates of each morphological stage. Then this proteomics data was put through few steps of data preprocessing to account for missing data and variability. And then the proteomic data in all the replicates of each stage was merged by their mean, and the enzymatic proteins catalyzing the reactions in Recon 3D were filtered out. Then this metabolic data was integrated in Recon 3D and using 3 different metabolic extraction methods, IMET, FASTCore, INET, which are independent of objective function, were used to build context specific metabolic model for each morphological stage. Now then the models were evaluated on the basis of metabolic functionalities, threshold independency and stoichiometric consistency, and we observed that the models built using IMET technique had the highest metabolic functionality and were robust to the choice of the threshold which were used during model building. So the model built using IMAT for each stage was further carried forward for the downstream analysis. Now, since we are not using any objective function in our study, so therefore we use GP sampler, which is a flux sampling technique for evaluating the optimal flux distribution. So the GP sampler generated a sequence of feasible steady state flux profiles. Among them, the flux profile with the maximum correlation with the input data was regarded as an optimal flux distribution. After flux evaluation, we found the reactions that were perturbed during the transitions and found that 675 reactions were perturbed during both the transitions. Till now, we have gathered the information about the metabolic perturbation involved during these phenotypic transitions. These perturbations are large in number, and targeting such large perturbation is not biologically feasible. Therefore, we developed a novel methodology named as reaction modulation, which cluster perturbed reaction on the basis of their combined effect on disease progression. So before reaction modulation, the correlation between the flux profiles of perturbed reaction was around 0.02 and 0.03 in both the transitions respectively, but after modulation it increased significantly. This suggests that the reaction clusters with highest correlation have great potential in progressing the disease further to its metastatic stage if they all are perturbed together. Further pathway analysis was done, and it was observed that the reaction in the top module of both the transition were involved in the transport pathways, TCA, pyruvate metabolism, and so on. And TAG pathway was predominantly involved in the top modulation strategy of transition 2. Now, to immobilize ovarian cancer progression towards its more metastatic stage, it is important to target metabolic perturbations that are involved in morphological transitions. So we performed in silico gene knockout using the genes catalyzing perturbed reaction. The genes essential for normal ovarian cancer cells were removed from our list as they cannot be targeted. So the non-essential genes were knocked out one by one in the target model and observed their effect in reversing the metabolic perturbation. The maximum correlation after the gene knockout increased significantly in both the transitions, and we observed that knocking down of SLC transport protein showed significant role in reverting back the perturbations during both the transitions. And also interestingly, we found that the genes involved in TAG pathway were also able to reverse transition to perturbations. So this follows our finding in the reaction modulation strategy that TAG pathway undergoes a significant amount of perturbation as it in reactions were predominantly involved in the top reaction modulation strategies of transition 2. So we next aim to investigate whether there are any available drugs that could be used for reversing the metabolic perturbation taking place during disease progression or not. So for this, we extracted the information of drugs from drug bank database, and then we approximated the optimal inhibition by the drug and evaluated their effect in the model. So we next came up with the list of putative drugs, among which were common to both the transitions, which are highlighted in blue. And osimertinib was also common for both the transition, and it was targeting NU1 gene. We also observed that the number of perturbed pathways associated with osimertinib was high as compared to other drugs, and the overlap value was also high as compared to the other drugs. So therefore, we chose this drug to experimentally validate our model predictions on the pharmacological regulation of both transition 1 and transition 2. So for in vitro validation, we added different doses of osimertinib ranging between 100 micromolar to 5 millimolar into the suspended OVCAR3 cells, and then cultured for 24 hours. And we observed that in this time period, control cells formed myeloid spheroids, whereas myeloid spheroid formation was impaired in the osimertinib added cultures, and similar was the case in the transition 2. So we next probed whether the impairment of transition 1 and transition 2 was a result of dose-dependent cytotoxicity exerted by osimertinib or not. So for this, the spheroids were treated similarly, but now they were stained instead for calcium AM and propydium iodide to detect cell viability and cell death. So we found that although disrupted, neither impairment nor integration of spheroids were accompanied by cell death. So we next asked if the spheroidogenesis disrupting concentration of osimertinib have any side effects on untransformed mesothelial cells. So we observed that no cytotoxic effect was observed. So therefore, this confirm our GSMM based predictions that osimertinib can be a potent disruptor of ovarian cancer spheroidogenesis without exerting any effect on the non-cancer cells. So with this, we conclude that targeting metabolic alterations during morphological transition of OVCAR3 impairs disease progression. So we are planning to take this study forward to evaluate the significance and robustness of our finding. So for this, we have thought to test osimertinib on other ovarian cancer cells, and if we get positive result, then this can be further taken up for in vivo validation. With this, I end my presentation here. I sincerely thank my supervisor and co-author for this study, my doctoral committee members, and THSTI, my home institute for funding, and my lab members for their constant support and help. Thank you.